Good morning, dear students. Welcome to this session of biology classes. Myself is Sumit, and today we'll be doing the 8th chapter, Human Health and Disease. In today's lecture, we'll be talking about the structure of antibody antigen interaction, and types of or classification of antibodies. In the previous session, we discussed about cell mediated and humoral immunity. In humoral immunity, antibodies are formed by the B lymphocyte cells of WBC, while in cell mediated immunity there is no antibody formation takes place. So let's start today's topic, and the first topic of today is the structure of antibody. Whenever you look at the structure of antibodies, the antibody is usually a Y-shaped structure. This Y-shaped structures have 4 chains. These chains are made up of peptide, and peptide means protein. So these are usually protein chains. Protein is polypeptide chain. So if you look at the structure, this one and this one, these two chains are longer in length, while this one and this one, these two chains are shorter in length. So there are 1, 2, 3, 4 chains, in which 2 chains are longer and 2 chains are smaller in size. The longer chains are known as heavy chains, while the smaller chains are known as light chains. So each antibody molecules has 4 peptide chains, in which 2 small chains are light chains, while the 2 large chains are heavy chains. So that an antibody is represented as H2L2. ये क्वेश्चन पूछा भी जाता है. Why antibody is called H2L2? H for heavy, heavy chain 2, light chain 2, heavy chain 2 and light chain 2. So the antibody is known as H2L2 molecule. It is always a Y-shaped structure. Whenever you see the structure of antibody, this Y-shaped structure has this region, this upper region, and the upper region has again of 2 parts. If you look at the upper region, this has also formed 2 parts or made up of 2 parts. This upper region, in which some zigzag manner structure is there, this upper region is known as V region, while the lower region of this antibody is known as the C region. So the meaning of V is variable region, and the meaning of C is constant region. So that the C means constant. This structure, up to this structure, all the antibodies structure up to this is basically same. There is no variation. All antibodies are similar, but the antibodies are dissimilar in this variable region. And this variable region, this V region, is the antigen binding region. As we know that the main function of antibody is to bind with the specific antigen and finally to destroy the antigen. So this variable region, this region and this region is different in each type of antibodies, while the remaining C region, constant region, is remain constant. It means the region is same in all the antibodies. So this one is the structure of an antibody. Now come to the next topic, which is the interaction between antigen antibody. So it is known as antigen antibody interaction, or sometimes it is also known as antigen antibody reactions. So now if you look at this topic, specific chemical interaction between antibodies produced by B cell. As we know that the antibodies are produced by the B lymphocyte cells of WBC, and antigens during immune reaction. Whenever there is a need of immune reaction, there is interaction between specific antigen with specific antibodies, and this chemical reaction or chemical interaction between specific antigen and specific antibody is known as antigen antibody interaction. The antigen and antibodies combine by a process called agglutination. The process of binding of antigen with a specific antibody is known as agglutination process. Each antibody is capable of binding only to a specific antigen. ये बात हम लोगों को पता है कि हर एंटीबॉडी स्पेसिफिक एंटीजन से ही बाइंड होती है. ऐसा नहीं होता कि एक ही एंटीबॉडी वन फॉर ऑल हो. इफ द एंटीबॉडी इज फॉर polio, so it can only bind with the antigen represented to polio, or attached with the polio virus, not to the other pathogens. So each antibody is capable of binding only to a specific antigen. The antigen determinant or epitope is recognized by the paratope of the antibody situated at the variable region. Variable region मैंने अभी आपको दिखाया था. सो देखिए, अगर हम सोचें कि दिस वन इज एन एंटीजन, तो एंटीजन का ये रीजन जो है, जो एंटीबॉडी से बाइंड होता है, इसे हम नाम दे देते हैं ईपीटॉप या एंटीजन डिटर्मिनेंट. ये है ईपीटॉप, और ये एंटीबॉडी के वेरिएबल रीजन से बाइंड हो जाएगा. ये वेरिएबल रीजन है ना, तो दिस वेरिएबल रीजन इज ऑल्सो नोन एज एंटीबॉडी बाइंडिंग रीजन. सो always the epitope or antigen determinant is recognized by the paratope. This region is known as paratope, the upper region. This region is known as paratope, and this from this paratope an antigen is binded. Let us suppose we again took a rough diagram. We again look at the rough diagram. This one is an antibody. The region, the upper region is known as FAB, and this region, the arm of Y or the arm of antibody is formed by FAB, and the stem of this antibody is formed by FC region. FAB antibody binding region, and FC is the recognition of target region. If you look at this particular structure, this one is the antibody, this one is the two arms are antigen binding arms, this one is the antigen, this region is FAB, and the FC region is recognize the target and attached to a specific cell like RBC. This is the receptor, RBC receptor, and from this receptor this FC region binds, and this FAB region binds to specific antigens. So this is about the types of the antigen and antibody interaction. Now come to the last topic of today, and this topic is the classification of antibodies or types of antibodies. Somehow some very important questions are put off from this particular heading, and very important for your competitive examinations as well as boards examinations. There are usually 4 to 5 types of antibodies produced in our body, in which the first antibody is IgA. Antibody is always a protein, a specific protein. This protein name is immunoglobulin. So the full form of IgA is immunoglobulin A, and immunoglobulin protein is there in each antibody. So if you look at the first antibody, which is known as the IgA antibody or immunoglobulin A antibody, it is usually a dimer. It means it is formed of two antibody molecules binded together. So it is usually a dimer. A rough diagram of this is this. This one is one antibody, this one is the another antibody, and these two antibody get connected, and these two antibody get connected to form IgA, immunoglobulin A antibody. So it is the dimer of two antibodies. It is usually the IgA antibody is present in the secretory glands or the secretion made by our body, like the secretion like saliva. So ये क्वेश्चन पूछा जाता है कि सलाइवा में कौन सा एंटीबॉडी प्रेजेंट होती है. तो इन आवर सलाइवा IgA antibody is present, except that the tears is also having some antibodies like IgA antibodies, the breast milk, the mucus, and these secretions have a rich amount of IgA antibody. This antibodies protect us against some pathogens. Then if we look at the next antibody, the next antibody is IgD, immunoglobulin D. It is the structure wise it is a monomer, it is not a dimer. It have a single molecule of antibody. It involved with the mast cells, and it is usually produced in the allergic reactions when there is an allergy of something. So for allergic reaction IgD एंटीबॉडी इज यूज्ड. ये भी क्वेश्चन पूछा जाता है कि कौन सी एंटीबॉडी एलर्जिक रिएक्शन में हेल्प करती है या एलर्जी को ठीक करने में हेल्प करती है. तो दिस वन इज IgD एंड IgE. द नेक्स्ट वन, the IgE, immunoglobulin E antibody, is also participate in the allergic reactions, and it also fight against the worms, the internal worms. तो इंटरनल वर्म से जो फाइट करने वाली एंटीबॉडी है वो IgE है. आप लोगों को ये बिल्कुल रिमेंबर रखना है, याद रखना है, क्योंकि ये बहुत बार क्वेश्चंस पूछे जाते हैं कॉम्पिटिटिव एग्जामिनेशंस में भी. नीट की आप प्रिपरेशन करेंगे या फर्दर कोई और एग्जामिनेशन की आप तैयारी करेंगे, पन्नागर की तैयारी करेंगे, BHU की तैयारी करेंगे, या JNU की तैयारी करेंगे, या आप नीट की तैयारी करेंगे, या मेडिकल से रिलेटेड किसी और फील्ड की तैयारी करेंगे, तो इनमें से क्वेश्चंस पूछे जाते हैं इम्यूनिटी से. द नेक्स्ट वन इज IgG. इट इज़ वेरी वेरी इम्पोर्टेंट. आपको ये IgA और IgG का ध्यान तो बहुत ज़्यादा रखना है. इट इज़ ऑलवेज ट्रांसफर्ड फ्रॉम मदर टू चाइल्ड थ्रू द placenta of mother. So this one is very very important, IgG antibody, because it is the inborn inherent antibody present in each child. मदर से डायरेक्ट ट्रांसफ़र हो जाता है बच्चे पे. मदर से जो बेबी है वो प्लेसेंटा से जुड़ा होता है, तो थ्रू प्लेसेंटा ही ये पास हो जाती है. और कौन सी एंटीबॉडी मदर से उसके चाइल्ड को मिलती है, तो दिस एंटीबॉडी इज नोन एज इम्यूनोग्लोबुलिन G, एंड इट इज़ वेरी वेरी एक्टिव अगेंस्ट एंटीजेंस. The last one is IgM, immunoglobulin M. It is usually the mix up of all the antibodies. Means IgM include IgA, IgD, IgE and IgG. One more thing you have to know that these in the structure of antibody, these molecules get connected by the two sulfur bond, or this one is known as this one. In all places this is connected through a disulfide bond. सो ये डाइसल्फाइड बॉन्ड्स के थ्रू कनेक्ट रहेगी. सो दिस इज़ ऑल अबाउट टुडेज लेक्चर. होप आप लोगों को ये समझ में आ गया होगा. अगर कोई आपको इससे रिगार्डिंग कोई क्वेरी होगी, कोई क्वेश्चंस होगा, तो आप मुझे इसमें कभी भी इस क्वेश्चंस को पूछ सकते हैं. इम्पोर्टेंट टॉपिक है एग्जामिनेशन के लिए. थैंक यू सो मच.